Hello. In this video, we will be introducing differential DNA extractions, including how they work and what they are used for. In order to discuss the purpose of differential extraction, we are going to introduce a case scenario. In this case, a 23-year-old female reported to the police that she had been sexually assaulted by a close friend. They collected the clothing she was wearing at the time of the assault, including this pair of underwear. The lab performed serological screening or presumptive testing and determined a semen stain is present. However, because this stain was found in her underwear, it is expected that there is a mix of her DNA and a potential perpetrator's DNA. How can we take advantage of biology to use this sample? In this stain, it would be expected to find the presence of both female epithelial cells mixed with the male sperm cells. However, the epithelial cells are going to be present in much greater proportion than the sperm cells, and the sperm cells are what we're after because we are looking for the perpetrator's DNA. Remember our presumptive testing for semen? One of those tests is going to prove extremely useful for ensuring that sperm is present in our stain. This is known as the Christmas tree stain. The Christmas tree stain is going to color the sperm cells' heads red and the tails green, while making them visible. The epithelial cells are going to hold only the red color. See how the epithelial cells can mask the presence of the sperm cells just based on numbers alone? This is where the differential extraction comes in. A differential extraction differs from a normal organic DNA extraction based on the contents of its lysis buffer. Remember, in a regular organic extraction, we are targeting all of the DNA, and we do a one-step lysis and purification where we put our 4 main lysis ingredients into the samples all at once, and then incubate and purify. In differential extraction, we are going to essentially conduct our lysis step twice. First, we use only SDS, EDTA, and proteinase K, which is going to preferentially lyse the epithelial cells, meaning the sperm cells are not going to be lysed. The second lysis step uses SDS, EDTA, proteinase K, and the addition of DTT, which is going to rupture the sperm cells. Now, why does this work? It works due to the way that epithelial cells and sperm cells differ in structure. Epithelial cells and their DNA-containing nucleus are held together simply by a normal phospholipid cell membrane and can be lysed easily by only using the SDS detergent. On the other hand, the sperm head, which contains the DNA, is going to have a type of bond that is much stronger than a normal membrane, the disulfide bond. These bonds cannot be lysed with SDS detergent and require an extra strong lysis reagent, dithiothriotol, also known as DTT, to break and release the DNA. The disulfide bonds are unique to sperm cells and are not present in epithelial cell membranes. Here's the complete workflow for differential extraction. First, lysis is conducted with only SDS, EDTA, and proteinase K, which is going to preferentially lyse the epithelial cells. Centrifugation allows the heavier sperm to pellet at the bottom of the tube, and the DNA from the epithelial cells is collected and put into a separate tube. This is known as the epithelial fraction. The second lysis step is then conducted on the sperm pellet using SDS, EDTA, proteinase K, and the addition of DTT, which is going to rupture the sperm cells. The DNA from this step is known as the sperm fraction. Once we have our two fractions, we continue to purify and concentrate both samples the same as we would in the regular organic extraction method before moving both fractions to the next steps in our DNA analysis: quantification, amplification, and separation and detection. While this method can be done manually by a scientist, it is very common in large forensic DNA labs to use automation. There are several benefits to automated DNA extraction methods. One, most importantly, being the ability to process more samples in the same amount of time. Automation also increases the precision of the extraction process. Automation involves an instrument with robotic components to complete the steps we just explained. These robots have a mechanical arm to move the samples around in pipette reagents and also contain a centrifuge to complete the centrifugation steps. There are two different types of instruments used for DNA extraction. The first are the QIAcubes, which are used for the lysis and separating out of the two fractions. We have a QIAcube and a QIAcube Connect. In our lab, we also have fun names for each instrument, and our QIAcubes are named Wally and Eva. The Connect is an upgraded version of the QIAcube. The easy instruments are used for the purification steps that occur after the lysis. We have an easy 1, Loki, and an easy 2, Thor, with the easy 2 being the newer version. In the extraction video we will show you next, we used Wally and Loki to complete our extraction and purification. Here we can see the inside of the cube. On the right, there are places to load reagents, pipette tips, and microcentrifuge tubes for sample collection. In the back left, you can see the centrifuge that is necessary to complete the differential extraction. The sample is loaded into a rotor adapter which has slots for tubes for the initial sample and allows waste to go into the bottom. After the lysis in the cube, the samples are purified in the easy 1. First, the magnetic beads in the reagent cartridges must be prepared for each sample through agitation and shaking. The beads are used for purification because they bind the DNA in a sample and allow the contaminants to be washed out. Next, the reagent cartridges are loaded into the slots in the instrument. The sperm and epithelial fractions have slightly different protocols. The sperm fractions are loaded straight into the easy 1. However, the epithelial fractions must have MTL buffer added first, as shown here. After the purification process is complete, the samples are removed from the instrument and are prepared for DNA quantification. Now we are going to look at the results of a full DNA analysis method in order to fully visualize the benefits of using a differential extraction for a mixed sample. Here we have the profile obtained from our sexual assault sample if differential extraction was not used. An individual person is either heterozygous, yielding 2 peaks from capillary electrophoresis, also known as CE, or homozygous, yielding 1 peak. So in a single source profile, we expect to only see 1 or 2 peaks at each locus. We can immediately see that there are way too many peaks at most loci in this profile. In a mixture like this one, we're going to see 3 or even 4 peaks at any loci within the data we retrieved from CE. You can see how this makes the data difficult to interpret and to distinguish the DNA profiles from the female versus the male contributors. In comparison to that last profile, we can see how nice the 2 profiles from the differential extraction look. In the sperm fraction, we see no more than 2 peaks at each locus, and the same is true for the epithelial fraction. This means that 2 single-source DNA profiles have been obtained. Here we have all 3 profiles lined up next to each other. You can see at each locus how the sperm and epithelial fractions separate the mixed peaks seen in the mixed profile. The separated fractions make it easier to perform comparisons and identify suspects with strong evidence backing up the results of any comparison. In conclusion, we have some key takeaways about differential extraction. Differential extraction is mainly used for samples from sexual assaults, where it is expected that there will be epithelial cells present from the victim and sperm cells present from the perpetrator. This would result in a mixed DNA profile if differential extraction was not used. The reason differential extraction works is because of the ability to preferentially rupture the epithelial cells versus the sperm cells using the reagent DTT. While manual extraction can still be used, many labs have switched to automated extraction in order to increase throughput. Finally, the benefit of the differential extraction is best visualized with the DNA profiles obtained, showing the separation of the male and female profiles, which makes comparison much simpler.